is in essence, so the simplest explanation that you can find is one where you make a model of only two transcription factors. So the idea is that in every stem cell, so if you have a stem cell, there's a fight going on. So in this particular case, this stem cell has two transcription factors. One is going to decide for a different cell fate than the other. But it's unclear who's going to win. So the network topology that's associated with it is that this transcription factor inhibits the other one, and vice versa. And as soon as it goes up in concentration, it makes more of itself. Okay? So clearly it's impossible to have high concentration of both. Yeah? So that's a symmetric situation that's unstable, because as soon as one of them wins, it will outcompete the other one. So it's only possible that this one wins or that one wins. Yeah? So if you plot the two transcription factors, concentrations as function of each other, then one possible, and these are the maximal concentrations, then one outcome is that it ends up in this state. So let's say transcription factor 1 wins and you get a brain stem cell that can differentiate further into neurons, etc., and astrocytes. And while this transcription factor, if transcription factor 2 wins, let's say you get a muscle cell. It's impossible to be in the state where they're both high, okay? So this is excluded. So that means that these transcription factors are continuously, say, having in a fight with each other, and as soon as one of them has won, well, the other one clearly has lost. But now, so how is that possible now? So even though we have a model now that indeed can only have two outcomes, so one this winner or that one is the winner. But how is it now possible that this stem cell, an offspring cell, is in the TF2 if one state, that one, or the TF2 state, that one? Okay? Well, that has to do with another effect that is not even in this network diagram, but all these reactions, all these molecular events that are hiding underneath these arrows, are in fact chance events. And I will explain that later to you. So all the molecular events that go on in a cell, if you would have two exact same copies of cells next to each other, different molecular events would go on in these cells at the same moment in time. So that means that this cell can go into the TF1 state, whereas that cell can go in the TF2 state. But which one will be in that state or in that state, that's something we cannot predict, because we do not have complete information about all the molecular events that go on in the stem cell. Okay, so it's these chance events on top of the network architecture, on top of the kinetic properties of these interactions, that allow us to give a molecular mechanistic explanation of cell differentiation of omnipotent stem cells into stem cells that have less and less capabilities to differentiate. Okay. So these are two fundamental examples in cell biology where it's easy to show that mathematical models have contributed a really important level of understanding of cell biology that molecular biology in itself was incapable of reaching. So it's really about the integration of network architecture with kinetics into a model that leads to predictions of cell behaviors. Those you can test against cell biological experiments. And if you do this long enough, then eventually you arrive at a satisfactory molecular mechanistic explanation of the cell hereby closing the gap between the genotype and the phenotype. So this ends my argument for why I think mathematical models are indispensable in cell biology. Thank you very much. See you next time.